Good morning. Sorry about the chaos in lecture this morning. I will hopefully get that technology sorted out by next Monday. I don't know what happened there. I totally practiced this in my office beforehand. Anyway, today I really just want to follow up on that pre-lecture video on sponges and talk about a few other topics with them. So as a reminder, the first quiz opens today on Wednesday, the 4th of September. Hopefully you'll have watched this lecture before you take that quiz. And just remember that on the 9th, Monday the 9th, we're starting normal labs, so it'll take the full time probably, and make sure to bring your forceps. And if you're on Zoom, you should probably get off because I am not on Zoom. Okay, so I just want to do a quick review of Porifera, then this is the outline for the rest of the lecture. So just to point out, remember poriferans, we're starting with them in the main lecture, although this is an online lecture now, a video lecture, but poriferans are not sister taxon to the rest of the animals, which has historically been what people have thought for many, many years, but just in the past 15 years or so, people have started to argue that ctenophores are sister taxon, and there's a lot of to the rest of the animals, and there's a lot of molecular data in support of that at this point, which is really confusing to have something so complex as a sister taxon to the rest of the animals. It really messes with our sort of ideas about animal evolution, as we talk about in that video. Okay. So the basic structure of these animals is two sheets of cells, and remember they are probably not epithelia, separated by mesohyl. So on the outside you have the pinacoderm out here, and on the inside you have the choanoderm. So this is the pinaco, and this is the choanoderm here. And in between those two things is the mesohyl, which is this region in here, and there are various cell types. There are like sclerocytes that make spicules. There are archaeocytes, which are just amoeboid cells that are walking around through there. There are also cells that make spongin, so those are called spongocytes. And I just want to point out that archaeocytes in particular, remember, can sort of wander all over the sponge. They can be in the mesohyl, they can crawl out to the outside, they can differentiate into something else. A choanocyte actually can differentiate back into an archaeocyte and wander around. So there's a lot of flexibility in the cell differentiation in particular in sponges. Okay, so the way this animal works is just that water gets pumped by those choanocytes so that it is pumping out of this osculum, this structure up here, this opening up here, and that draws water in through here, through these ostia, which there are many, many of them around the sponge. And as the water passes by those choanocytes on the inside of the sponge, food particles are captured from there, right? This is a very simple sponge. It's an asconoid sponge, but as I pointed out, there are 3 grades or levels of complexity. And for me, I remember them as ASL, which is the acronym for American Sign Language. Asconoid are the simplest, syconoid are a little bit more complex, and leuconoid are the most complex. And most living sponges are leuconoid sponges. But, they're all the same in the sense that choanocytes are powering water flow through these. It's just that there's a much larger surface area of choanocyte or choanoderm in a leuconoid than there is in an asconoid because of all the folding and chambers. Okay, and we talked about how as water passes by those choanocytes, food particles are trapped on that collar of microvilli around the flagellum and then get carried down into the cell where they're phagocytosed. Okay, there's just a nice illustration of that, and then they can be used by that choanocyte or they can be transferred to another cell. There's a lot of movement between cells and movement of cells in sponges. Okay, let's talk about a few more topics if I can get out of this video. Okay, I want to talk about 2 phenomena related to flow, like we just talked about, flow through sponges. We just covered the basics. Sponges really are just basically plumbing systems, active plumbing systems, and there's a bunch of interesting topics related to how they generate flow through their bodies in sponges. So let's talk about 2 of those. Okay, so first of all, choanocytes are generating flow for suspension feeding, right? And here's a nice leuconoid sponge, and you can think of this, as we have already, as having water entering in all of these ostia throughout the body here. And there's many, I'm not labeling them all. And water is exiting through the osculi, of which there are 2 labeled on this individual. There might be more, there might be fewer, depends on the animal. Okay, so here's a basic concept that you will understand. These red arrows represent water coming in. there's a total volume of water that's coming in through all of these ostia. And that total volume of water has to exit the sponge at the same rate, right? There's no loss of water in between. So one way you can think about this animal is like, imagine a water hose, like a garden hose or something like that. And the different chambers can be represented in that hose just of different diameters. So for example, if you sum up the cross-sectional area of all the ostia, so let's get back here. Okay, so look at this little ostium, this little ostium, this little ostium, this little ostium, etc. That adds up to a cross-sectional area part of that hose. So here is a representation of a sponge as a single hose, and this is the ostia cross-section. People have actually measured this in sponges. We'll return to some actual measurements in a minute. And then that water makes its way into the choanocyte chambers, and you can call those choanocyte chambers another part of the hose. The cross-sectional area, the total cross-sectional area of choanocyte chambers is huge in sponges, so it's much bigger than this. So that hose all of a sudden has expanded dramatically. And then the last part of the hose is these osculi. And the osculi tend to be quite large, like these two, but there are very few of them. So the total cross-sectional area of those is very small. So that part of the pipe is osculum. And this part is choanocyte chamber. Okay, so as I mentioned, so this is like relative, just like a cartoon of what this water flow system might look like, but people have made measurements of the cross-sectional areas of these for given individual sponges, actually usually small chunks of sponges, because this is very painful work to do, and one of those estimates is here. So the ostial cross-section, so we're talking about this area right here, was 3.14 centimeters squared for this one particular sponge. The choanocyte cross-sectional area, so we're talking about this cross-sectional area right here, was 203, I think, centimeters squared. Yes. And the osculum cross-section was 0.03 centimeters squared. Okay, so we're talking about a single garden hose, but just it has regions that are very big in diameter and regions that are small. You can imagine what happens if you pump water in at a given flow rate into here, which is what's happening in that real sponge. It's coming in at a certain velocity, but then as the hose gets much wider, once this water enters those choanocyte chambers, it slows down because that hose is so much bigger. And then as it enters the osculum part of the hose, it's so narrow that it speeds up. So it's like a jet there. So you can make estimates, and you can actually measure this, or you can just estimate it from fluid dynamics, what those velocities of water should be. So let's just draw a corresponding graph down here. So this is the ostial velocity, this is the osculum velocity, this is the choanocyte chamber velocity, and here's flow velocity, and we'll do centimeters per second here. It's going up here. Okay, so what that looks like is if water's coming in at a particular flow velocity here, it hits the choanocytes and it drops down to very little flow velocity, and then once it hits the osculum, it speeds up dramatically, just because of the diameters of the hose, right? Okay, so people have measured those. This is 0.06 centimeters per second for a given sponge. This is 0.0008 centimeters per second, and this is 5 centimeters per second for the osculum area of that sponge. Okay, so this is the principle is just that there is continuity of flow. What goes into the inlet of a sponge, aka all those ostia, the amount of flow that enters there has to be, per unit time, has to be equivalent to the amount that comes out of the osculum per unit time. And that means the flow velocity of water in those different compartments varies just based on the total cross-sectional area of those compartments. Okay, who cares? This is really interesting for sponges for two reasons. So one is this. The flow velocity in the choanocyte chamber is super slow, water spends a lot of time in choanocyte chambers in sponges, and what that means is that choanocytes have a lot of time to remove particles from that water. So they are very efficient at removing particles from water. Okay, so if some water enters a sponge, when it exits that sponge, there's going to be very, very few particles left in it because the sponge will have captured most of those. In fact, they capture not only things that we think of as particles, but they also can capture dissolved organic material. So amino acids and small proteins and things like that dissolved in water, they can remove. And that efficiency is largely because water is sitting there for a while, getting filtered by multiple choanocytes. Okay, so that slowness in the choanocyte chambers is what leads to that property. If you remove all the food from a given parcel of water, then you really, really don't want it near you again, because you might suck it back in and there's already no food in it. So if you're a sponge, you might want to throw that water far away from you when it exits, and that's what this is. So they eject used water with a high velocity flow, like a squirting attachment at the end of your hose or something like that. Okay, so slow flow in the choanocyte chambers leads to very efficient removal of particles, and high flow at the osculum leads to them shooting that used water far away from themselves, where hopefully they will never see it again. They will never take it into their bodies again because there's nothing in it anymore. Okay, this is all just continuity of flow. Okay, so their bodies are sort of arranged to take advantage of this. Okay, I have another flow-related topic which is really interesting as well, quite different from this one. Okay, so that has to do with the generation of flow through the body, which we've already talked about a little bit. And so this is based on an observation made in the 1970s by a guy named Steve Vogel, who's a biomechanicist, and he made the observation that a lot of sponges look like this. They're attached on the bottom, but the osculi tend to be up high. So here are the osculi on this sponge sticking up away from the bottom. Here are the osculi on this sponge sticking up away from the bottom. Likewise, these look like little tiny volcanoes on this purple sponge here. Okay, and he wondered why that might be, and he started thinking. He was a fluid dynamics person, so he had some ideas about that. And let me just draw a schematic of that so you can see what it looks like. So if this is the sea floor down here, this could be a freshwater sponge too, of course. So it could be the floor of the bottom of a river or a pond or something like that. So the sponge, or a sponge, might look something like this, like a little volcano on it. So it has a single osculum at the top. It has all these microscopic ostia covering the whole body. That's where water is entering. So you can see the ostia are lower than the osculum. They're further away from the substratum than the osculum. The osculum is further away from the substratum than the ostia are. Okay, so Vogel had some ideas about why that might be, and he thought that was related to environmental flow. So any aquatic animal is in a current at all times. There's really basically no still water. It may be slowly moving, but there's no still water really in the ocean or even in a pond or a lake. So let's just represent that by an arrow here. So this is going to be environmental flow, aka a current, a water current. And the length of that arrow represents the velocity of that current, and the direction is given by the way the arrow is pointing, obviously. So it's moving from left to right in this video. Okay, so he thought that the flow through the sponge might be related to the strength of that environmental flow. So he wanted to measure flow through the sponge, so he measured this flow as well as environmental flow, which was osculum flow. Like, what's the rate of water leaving the osculum? What's the velocity of water as it exits the osculum? Okay, so he went out and did this for a bunch of sponges in the Caribbean. He also did it in the lab where he could manipulate the flow in a flow tank. And he made this amazing discovery, which is this. If you plot environmental flow velocity here, so in terms of centimeters per second, for example, it's increasing in that direction, against osculum flow, which I showed you where he's taking that right at the osculum, increasing this way, also centimeters per second. We're not going to actually use numbers here, but you'll see the pattern. and you measure a single sponge in many different environmental flow velocities, you get this amazing pattern of oscular flow, and it looks like this. There's a little noise. It's the world out there, right? Okay, so what it looks like is that, and if you draw a regression line through it, it explains like almost all of the variance in those lines, in those points. They're almost all basically on that line. So oscular flow is determined in part by the current around that sponge. Okay. All right, so this is a very peculiar correlation, and he had a variety of ideas for why this might be, and normally I would ask you about those, but I'll just point out, well, let me just point out one of them, actually, that's incorrect. One of them might be this, okay? We know that sponges generate flow through their bodies using choanocytes, right? So one of the ideas for why they might pump more water through their bodies at higher water currents, higher environmental flows, is that at higher water flows, maybe there's more particles coming by them, so they say, oh, there's a lot of particles in this water, let's beat our choanocytes faster. It might be an active response on their part. So Vogel actually tested this, and he did it in a very simple and clever way. I should point out that this is for a live sponge, this line. So what he did was take that same sponge he'd measured that on, and he killed it. He put it in fresh water. These were marine sponges, so fresh water killed them, and then he put them back in currents to see what would happen. So he tried to see if this held for dead sponges as well. And it did, except not exactly the same way, but very similar. So for a dead sponge, the same dead sponge, he found a line that looked like this. So in a dead sponge, the oscular flow is also related to environmental flow, so it can't be an active response of the sponge. Obviously, it's dead. Okay, so that was really interesting. So what was this? So what he concluded that it was a feature of water flows, or actually fluid flows, in any situation when fluid flows are flowing over a surface. So let's look back at that drawing on the left of the sponge on the substratum. We have this environmental flow up here of some magnitude, 1 meter per second or something like that. Who knows? That's a pretty fast current. Okay. What is the water flow right down here on the surface of the bottom of the ocean? Let's assume it's perfectly smooth and everything. How rapidly is it moving? It is not moving at all. So the velocity down here is 0. That is called the no-slip condition. When a fluid flows over a surface, right at that surface, it is not flowing. It is stuck to the surface. The water molecules are actually, stuck onto that surface. Okay, so up here you have pretty high velocity, and up here you have no velocity. And so what happens is that no velocity at the bottom exerts friction on the layers of water above it. So what you get between those two is a gradient of velocity. So it's a little bit slower right there, and a little bit slower there, and a little bit slower there. So as you go towards the surface, water flow gets slower and slower. Okay, so that's a velocity gradient in that environmental flow. And that, you guys may have heard of this before. This is what is called the boundary layer. So there's always a boundary layer over any surface, unless the flow is very turbulent or something like that. Okay, so that means that this sponge is immersed in a velocity gradient. And in particular, what it means is that flow going by the osculum is at that velocity, and flow going by the ostia is at a lower velocity. It's slower water down there. And we know from Bernoulli's principle that the pressure within a fluid is related to the velocity of that fluid inversely, right? So at low velocities, the pressure is high. So down at the level of the ostia, there's high pressure, and up at the level of the osculum, there is low pressure, just because of Bernoulli's principle. So think about how an airplane wing works, and once you remember that, you'll see that this is just like an airplane wing full of holes, basically. So it's low pressure up at the level of the osculum just because of the velocity of that environmental flow. Because of that, you automatically get flow driven from this area of high pressure to an area of low pressure. So you can take a dead sponge and put it in an environmental flow, a water current, and as long as it's attached to a surface, and there will be flow passively induced through that sponge. Okay, so they're not expending any energy to generate that flow. It's just driven by that Bernoulli's principle, because they're in a flow gradient. The ostia are in a higher pressure area than are the osculi, and water is blown out of that. Okay, so that is really cool. That's not the only animal that does this. Think about a keyhole limpet or an abalone. They have holes near the top of their shell. Water gets pulled over their gills passively because of that, if they're in a water current. There's lots of other examples of that, like prairie dogs, for example. Not a marine invertebrate, but whatever. They dig holes in the ground. They have a pile of dirt up at the top that makes a little volcano. As the wind of the prairie blows over that, it pulls air out of their burrows, and thus it ventilates their burrows. So lots of examples of this, not just in water, but also in air. Okay, we'll see it again in the class. All right, so that's two completely different flow-related phenomena with sponges, and altogether it just sort of contributes to their amazing ability as filterers of seawater. And I just want to point out something I just saw in the literature. In the past 2 years, people have started using sponges as natural samplers for environmental DNA, because they move so much water through them, they capture so many particles, that people are starting to think, wait a minute, everything is passing through a sponge. Maybe we could use these as samplers for environmental DNA. And indeed, not only can you do that, like in a given ecosystem, go out and collect some sponge, like here's a paper that did it in the North Atlantic in the deep sea. But if you collect a sponge and preserve it, like in a museum, for example, you can later go back and look at the DNA that's preserved in that sponge. So people are even using museum specimens as sources for historical environmental DNA, which is really amazing. I just saw that when reviewing the sponge literature a couple of days ago. Okay, so let's talk about some other sponge-related things. Defenses, for example. We already talked about spongin being a defense. It makes it hard to chew on a sponge that has spongin. Not all of them have spongin. And spicules are a pretty good defense as well. It's hard to bite because, you don't want to bite some sharp glass thing. It might cut you up. And there's certainly some animals that specialize on feeding on sponges, like some sea slugs, for example, hawksbill turtles. There's a few fishes that do as well. But most animals are, don't really want to be trying to saw through spongin or trying to bite a whole bunch of shards of glass or shards of calcium carbonate, depending on what kind of skeleton it has. But they also have other defenses in addition to these obvious ones, and mostly what I want to talk about are chemicals, because sponges make a ton of bioactive secondary metabolites. These are just metabolites that are not involved in their own metabolism. They're a product of their own metabolism that they then store in their bodies. And they use those for a whole bunch of different purposes. So some of them are anti-predator, some of them are anti-fouling, and so what that means is that the larvae of other invertebrates or algae settle on a sponge perhaps, and then they die because of this secondary metabolite that's stored there. They might use them to compete for space with other organisms, and you can see that in this photograph down below. So here's a coral, here's a sponge, and you can kind of see the sponge, there's like this zone right here. This is actually dead coral. The sponge is advancing, and as it's advancing, the coral is dying, and then the sponge can grow onto that, and then the next bit of coral dies. That's because of some chemical that the sponge has in its tissues. And then, of course, they use them to control bacterial growth as well, and perhaps other pathogens as well. Okay, this is actually really important to humans because sponges make so many bioactive secondary metabolites that people prospect in sponges for chemicals that might be useful in fighting cancer or other things. And so there are numerous approved drugs from that were originally derived from sponges, and there are many more in trials. Let me just point out one key one. That is this drug called cytarabine, which is a modified version of a sponge chemical that was called spongothymidine and spongouridine. So two different chemicals taken from this sponge, Cryptothecia crypta. So these are nucleoside analogs, but they don't have deoxyribose as a sugar. They have arabinose. So what happens is that if cells, eukaryotic cells, are dividing and they're exposed to these analogs, they take it up, they put it in their DNA, but they can't be translated into protein, so eventually that cell dies. Okay, so this would be very useful, of course, to attack cells that are dividing very rapidly, for example, cancer cells, and so that is indeed what cytarabine is used for. It's for leukemia, and it's one of the World Health Organization's essential medicines because it's so important in treating leukemia. Only one of numerous others. Okay, where do those secondary metabolites come from? Well, either the sponge makes them or something that lives with a sponge makes them, and most of the time we don't actually know the answer to that question. It turns out the sponges have lots and lots of microbial symbionts, so lots and lots and lots. And so mostly we're talking bacteria here, but also archaea, also fungi, and in some cases the microbes make up almost half of the biomass of what we're calling a sponge, but really it's half microbe, half actual sponge tissue. Okay, just in the past 15 years or so, people have started to realize that you could categorize sponges into two relatively discrete categories as LMA or HMA sponges, low or high microbial abundance. So it's pretty discrete. Here, some have relatively few and some have buckets of them. And then the amazing thing is that there are differences in the biology of these sponges related to their microbial abundance. So for example, HMA sponges, high microbial abundance sponges, have longer, narrower pipes in that whole system for moving water around, and they pump less water through there than do low microbial abundance sponges. No idea why, but definitely true. But just like an interesting pattern that may have some, biological significance. We just don't understand what it is at this point. Those microbes, in some cases, definitely produce some of those secondary metabolites. Some of them are photosynthetic, like some cyanobacteria, and those may provide photosynthate to the host. Some of them fix nitrogen, again, often some cyanobacteria, but others as well, so they can supply that to the host. Like in some situations, like in coral reefs, that nitrogen in usable forms is pretty uncommon, so very useful. And then, of course, so those microbes are not always good. They can be bad as well and pathogenic to the sponge. Okay, let's see. So that's symbioses. Let's just talk a little bit about their ecological roles, which there's tons of ecological roles. There are things that eat them, of course, as I mentioned, but I'll just point out a few that are of particular interest. So one of them is that in systems that have a lot of calcium carbonate, like, for example, think coral reefs, sponges are extremely important in both forming reef structures, and then different species are important in eroding reef structures. So in particular, there's a lot of boring sponges, which does not mean they're uninteresting. It just means they bore holes in things. Like, for example, this is an oyster shell with all those little yellow marks on it. Those little yellow marks are a boring sponge, a little bit sticking out, but really there's tons of sponge inside that shell. Really weakens it. They're chipping away at the calcium carbonate, so they're turning that shell, or they're turning a chunk of coral reef into sand. And they do this in pretty complicated ways, but they're really important on, a given square meter of coral reef. Boring sponges are removing something like 1 kilogram or 2 of calcium carbonate from that reef every year. So they're actually degrading. The reefs are constantly being degraded by these sponges. So in order for a reef to be maintained, it has to also continually be adding calcium carbonate, which we'll talk about in a few weeks. Okay, the sponges that have photosynthetic symbionts may be net primary producers, or if they have nitrogen-fixing symbionts, they might be producers of organic nitrogen, making them important in a system. And I just want to point out one other thing that has to do with coral reef systems. This is a hypothesis that people came up with just a few years ago called the sponge loop. And it relies on, sort of a recognition of the importance of sponges in retaining dissolved organic material, nutrients in particular, in coral reef systems. So the idea is this. Coral reef systems are, bright light, typically speaking, shallow water coral reefs. And so things that are photosynthetic there, corals and algae, produce a lot of photosynthate. In corals, what that often results in is the corals release a ton of mucus into the water. Algae release some photosynthate as well into the water. But those are dissolved organic compounds, and they are at risk of being carried off the reef into the open ocean and lost to that reef structure. But that dissolved organic material can be taken up pretty efficiently by sponges because of that the long residence time of water in choanocyte chambers. So that DOM can be taken up by sponges, and they put it into their bodies, and then sponge tissue sort of flakes off, cells flake off. That's particulate organic matter. Other things can eat that. So they're holding on to dissolved organic material that otherwise might be exported off of a reef system. And we don't really know how important that is at this point, but it's potentially very important for coral reef systems. Okay, reproduction is really interesting in sponges, and I just want to cover one aspect of it in detail. Sponges are really great at regenerating from fragments. You've even heard stories, I'm sure, of people taking sponges and passing them through a mesh and breaking them up into individual cells, putting that in a container, and those cells may crawl back together and actually form an intact sponge. That only works in a few species, actually, but all of them are pretty good at regenerating from fragments of the sponge. I want to point out one way of reproducing asexually that many freshwater sponges do, and very few marine ones do as well, and that is seasonal production of what are called gemmules. Okay, so this is really easy to explain in freshwater sponges. It's harder to understand in marine sponges. So let's talk about freshwater sponges here. So what gemmules are just like a little container. This might be like maybe 250 microns in diameter, like a little bead basically. That's a hollow sphere of collagen, spongin, and spicules embedded in that, and inside that a bunch of archaeocytes. Okay, so those are just undifferentiated amoeboid cells, and so they form these. So this is one gemmule. Freshwater sponges form these just before the season gets really bad, and so these little, the adult sponge may die, but these can overwinter. They can survive that season. I'll walk through that in a second. And then as soon as conditions get better, these cells can crawl out of this little opening and reform a new sponge. So here's a quote-unquote hatched gemmule right over here, and around it is that new sponge developing. Here's an osculum right up here. And here's another one right over there. Okay, so what does this look like when they do this in fresh waters? It looks like this. So here's an adult sponge actually that's kind of dead probably, but all of these are little gemmules that are produced. So this freshwater sponge produced a ton of gemmules. So what's the situation? So what happens is this. In the summer, there's a lot of food. Day length is long, it's, the temperature is relatively high. It might be a great time to be an adult sponge. So in the summer, we have adults that are happy. That's a slight oversimplification, a non-scientific term, but whatever. They have food, everything's good. Okay, so maybe it's turning into fall. Temperatures are getting cooler, there's day length is shorter, so there's less photosynthesis, so maybe there's less food, lower temp, and the adult sponges realize, quote unquote, anthropomorphizing. Of course, this is an evolved behavior. They realize that soon they will be dead in the winter, and so what they do is they make gemmules, like the adults on the right there. Gemmules. Okay, then in the winter time, it's terrible. Maybe in the northern hemisphere, in whatever hemisphere winter, your pond gets completely covered with ice. There's no food, so there's no food. It's very cold, and then the adults die, but the gemmules remain. The gemmules are just sitting there at the bottom of this pond, and then what happens in the spring is that by some mechanism that we don't understand, so food and temperature are going back up again. By some mechanism we don't understand, those archaeocytes inside the gemmule start to crawl out, and they form this new sponge. This is a clone of its parent, right? It's just asexually produced from cells of its parent. So that's a form of asexual reproduction over seasons, right? And it's a way of surviving over a challenging season. So we usually call these things overwintering devices, overwintering structures or devices. And we'll see some other ones in this class. It's particularly common in freshwater because the seasonal swings are much bigger than they are in marine systems, right? So in marine systems, these are not that useful typically. Okay, so that is one form of asexual reproduction. Sexual reproduction in sponges is very complicated, and we actually know very little about it, but I'll just point out that Most of them are hermaphrodites. So they're usually sequential hermaphrodites, males first, then females, but sometimes simultaneous. There are a few that are separate sexes as well. The gametes develop from archaeocytes, or they develop from choanocytes that dive back into the mesohyle and become archaeocytes. So eventually they form from archaeocytes, sperm or eggs, or both, depending on the group we're talking about. Okay, males free spawn their sperm into the water. So here is a sponge, at least acting as a male. There's the osculum up here, and this cloudy situation is a bunch of sperm. Some other sponge of the same species nearby some other sponge nearby will catch that sperm. They have choanocytes. They're perfectly ready to catch tiny things, right? If it's a sponge of a different species, it'll just eat the sperm. If it's a sponge of the same species, what happens is the sperm get caught by a choanocyte, they get phagocytosed as if it's food, but then the choanocyte moves into the mesohyle and transfers the sperm to an egg, which is crazy. So you have these intermediate cells, choanocytes, that grab sperm and move them to the target cell, the egg. Okay, so that's very peculiar. That's really poorly known in sponges, but it's been described a few times. Then that egg develops in the mesohyle and eventually gets released as a non-feeding larva, and those non-feeding larvae are super diverse. But so here's an example of some larvae that were brooded inside this sponge, and now we're swimming out the osculum of that sponge. This is a local sponge called Leucoselenia. Okay, and then, let's see, let me advance. All right, so development in sponges is very complicated. There's lots of different kinds of cleavage patterns, and they're just messy. Blastulae are complicated. Gastrulation, we don't even know if they undergo gastrulation, generally speaking. Remember, we talked about gastrulation in that form and function video earlier on. And I just want to point out one really interesting difference between sponges, the few times they've been looked at, really only once actually, and the rest of animals. So if you remember from that video that on form and function, we talked briefly about embryonic development, cleavage, formation of a blastula, so blastula being often a hollow ball of cells, and then that blastula going on to form a gastrula, so in-pocketing here. So this is a gastrula. That process is called gastrulation, and then after subsequent development, you end up with an animal that's like a tube within a tube with these two layers. Let me just point out that this inner layer we usually call endoderm, and I'll just show you here. The endoderm of a gastrula forms most of the digestive system of an adult. So this is really for a cnidarian or a bilaterian. So almost all animals do basically this. Okay, so do sponges do this? So we can find some sponges that make perfectly good blastulae. So let's look at a sponge here. Perfectly good blastulae that do something that looks very similar to gastrulation. So they make those two layers, and we can call that endoderm here. But then what happens when you go on to form an adult? Because, you know, if you look at a sponge, a simple sponge, it is kind of a tube within a tube, right? The outside tube is lined with pinacoderm, the inside tube is lined with choanoderm. So does it do the same thing that the cnidarians and bilaterians do above, that inner part of the gastrula, the endoderm forming that inner part of the sponge? Well, if you look at a like Leucoselenia type sponge here, it looks like this, and there's that inner layer that has choanocytes. I won't draw the ostia through here, just like our generic drawing of a sponge, right? Here's the seafloor down here. So we would expect if this was similar, gastrulation in a sponge was similar to gastrulation in an cnidarian or bilaterian, that those green cells would be on that inner layer. But in fact, if you track those green cells, they end up all over the place, inner layer and outer layer. So gastrulation in sponges looks pretty different from gastrulation in other things. So sponges are strange animals. I should have just started with that. That's really the point of this lecture. They're quite strange. Okay. Couple more things. So here's a paper that described that. They labeled cells of the endoderm, and those cell layers established during during embryogenesis had no relationship to the cell layers of the juvenile sponge. Okay. Only thing I want to talk about now is sponge diversity, which is actually pretty simple. Two big groups within the phylum Porifera, and those are the Calcarea and the Silicia, and those names tell you quite a lot about them. So calcareous sponges have calcareous spicules, siliceous sponges have glass spicules. All siliceous sponges have glass spicules. Okay. Within the Silicia, there's two groups, the demosponges and the Hexactinellida. We think that this taxonomy here, roughly reflects phylogeny. So demosponges and Hexactinellids are sister taxa, and sister taxa onto that is the Calcarea. Okay. Calcareous sponges can be asconoid or cycanoid or leuconoid depending on the species. They always have calcareous spicules. They don't have any spongin. Siliceous sponges within the demosponges, almost all sponges are demosponges, close to 90% of them, and they're all leuconoid. Okay, so if I ever point to some unidentified sponge and ask you, is it a Hexactinellid, a demosponge, or a calcarea? You should pick demosponge if you don't know anything, because that is what most sponges are. Also, if I point to a sponge that you know nothing about and I ask, is it asconoid, cycanoid, or leuconoid? You could just say leuconoid as a default because almost all sponges are leuconoid. Okay. So demosponges have glass spicules, leuconoid. Most sponges you'll encounter in the ocean are demosponges, or in fresh water for that matter. The last group is the Hexactinellida, and that is a very, very strange group. They have 6 pointed spicules, so spicules that have 3 crosses, basically, so 6 tips. They don't make spongin, mostly deep sea. They're mostly pretty uncommon, although there are big reefs of Hexactinellids in the deep sea as well. It's actually kind of impossible to assign them to asconoid, cycanoid, or leuconoid because of one thing. They are syncytial. So that is super strange. That means that they're composed of just a few huge multinucleate cells. And it's basically impossible to understand their structure unless you're like a specialist, of which there's really only 1 or 2 people in the world. So one of them is a woman named Sally Lees at the University of Alberta, and she calls Hexactinellids large multinucleate amoebae strung out as a cobweb over a skeleton of spicule. Okay, which is it's hard to understand what that means, honestly. Okay, so here's a group of Hexactinellids with next to that crab right there. And I'll just point out one that you've seen before, the skeleton of at least. So this is Euplectella, which is called Venus's flower basket, which is this deep sea Hexactinellid that anchors itself into the substrate, into soft sediments, by these long glass fibers like this. There's sort of like roots, and then the animal skeleton sticks up like that, and then of course there's multinucleate cells sort of draped over that because it's a Hexactinellid. And you guys all know this as well because they host usually a pair, sometimes more, of shrimp inside, which are called glass sponge shrimps. These go in when they're larvae, and then as they get bigger, they get stuck in there. So they're trapped. Usually it's a male and female. So in Japan, in particular, this is a symbol of undying love, these Hexactinellids, and people give skeletons of them as gifts. You could also view them as, a symbol of prison, but, a slightly different take on it there. Okay, I just want to point out one twist here to sponge biology. We've spent all this time talking about how amazing water flow is in sponges. Well, there are sponges that don't do water flow at all, it turns out. They don't have choanocytes, they don't have any ostia or choanocyte chambers or osculi at all. So those are predatory sponges, and these have been really worked on in the past 25 years or so. And these are sponges that are usually pretty small, but there's some very large ones as well. They're mostly in weird habitats like the deep sea or shallow water marine caves. The small ones look like little plants, so they look like a little stalk here and then a bulb at the top up here. So let me just, and the big ones work the same way. So let me just explain how these work. In the bulb part at the top, they have a bunch of spicules. They're perfectly normal sponges. They're demosponges. And those spicules are hooked things. So they're like the hooked part of Velcro, that side of it. And so what happens is that little crustaceans and little, larvae and those sorts of things swim by. they get caught in those hooks. So here's a little crustacean that got caught in the hooks of one of these predatory demosponges, and you can see that here. Here's a little crustacean over here. So what happens, there's no mouth or anything. What happens is that the cells of the sponge then migrate over and start digesting it. So here is this same sponge as in A over here. I actually don't remember how much later, like a day or two later, and that crustacean is in here just buried by sponge cells that are now breaking it down. Okay. So they capture small swimming prey. They're suspension feeders. This is a tiny one. We have some big ones off of our coast. This is a harp sponge. I think this is like 2 meters across or something like that in deep water in the Monterey Submarine Canyon. The thing I just want to point out is that, sponges are these nice simple body plans organized around water flow, but even with that super simple body plan, you can do something completely different, and that's what these predatory sponges are doing. Okay.